Okay, my name is Matthew, and I'm going to be discussing the regulation of sperm maturation in the germinal epithelium. So I'd just like to give a little overview of the male anatomy. The anatomy of the male reproductive system can be divided in terms of the external and the internal genitalia, and the gonads, the testes. The external genitalia consist of the penis and the scrotum. The internal genitalia consist of ducts and accessory glands, including the seminal vesicles, the urethra, the prostate, and the bulbourethral glands. The location of the testes and the scrotum is significant, as spermatogenesis occurs at a lower temperature, about between 1 and 8 degrees Celsius lower than the average body temperature, in order to produce viable sperm. This presentation will focus on sperm production and maturation. Sperm is produced in the seminiferous tubules inside of the testes. The seminiferous tubules make up about 80% of the testicular mass in an adult male. Sperm matures in the epididymis, where it is stored until it travels through the vas deferens and eventually the urethra during ejaculation. This next slide focuses on the seminiferous tubules, which is the primary site of spermatogenesis. As I mentioned, spermatogenesis occurs in the seminiferous tubules inside of the testes. Spermatogonial stem cells begin in the outermost section of the seminiferous tubules, adjacent to the basal lamina of the tubule walls, and they mature inward toward the tubule lumen, where eventually immature sperm reside. Peristalsis of the smooth muscle, similar to that of the digestive system, pushes spermatocytes towards the tubule lumen. Columns of developing spermatocytes develop between Sertoli cells, which link to one another via tight junctions. This basal lamina, along with these tight junctions, compartmentalizes the seminiferous tubules into the tubule lumen, the basal compartment, and the interstitial fluid, each of which have different chemical compositions to support sperm development. Leydig cells are one of the two important cell types in the seminiferous tubules, along with the Sertoli cells. Leydig cells are dispersed throughout the interstitial compartment and secrete testosterone into the tubule. Sertoli cells support spermatogonia development by providing essential nutrients. They produce inhibin and activin, in addition to androgen binding protein, or ABP. ABP binds testosterone secreted by Leydig cells, preventing it from diffusing out of the tubule lumen. And this is just a little graphic depicting the seminiferous tubule with the lumen on the inside and the basal lamina on the outside, with the spermatogonia maturing inwards towards the lumen. So spermatogenesis occurs in that spermatogonia are germ cell precursors to mature sperm. These spermatogonia reside just inside of the basal lamina of the seminiferous tubules, as we just saw. Spermatogonia undergo mitosis, in which one spermatogonium splits into two identical daughter cells, all containing 23 pairs of chromosomes. In order to not deplete the store of germ cells, some cells enter meiosis to develop into primary spermatocytes, while others remain spermatogonia to continue creating germ cells. Each stage of differentiation brings sperm cell precursors closer to the lumen of the seminiferous tubule. The primary spermatocyte undergoes meiosis 1 and divides into two secondary spermatocytes. In this process, each subsequent cell receives half of the chromosomes of the parent. The primary spermatocyte has 23 pairs of chromosomes, producing two secondary spermatocytes with 23 chromosomes each. In meiosis 2, the secondary spermatocytes differentiate into spermatids. 2 spermatids per secondary spermatocyte produces 4 spermatids. Each spermatid has 23 single copies of each chromosome, half of a complete set of 23 pairs of chromosomes, the egg having the other half. The spermatids then differentiate into mature sperm cells, also known as spermatozoa, during the process of spermiogenesis. So I'm just going to discuss a little bit the hormonal control of the spermatogenesis process that I just discussed, spermatogenesis follows the model of the classic endocrine hypothalamic pituitary axis, mediating communication between the hypothalamus, the anterior pituitary, and the testes. Gonadotropin-releasing hormone, or GnRH, is a tropic hormone released by the hypothalamus, which in turn controls secretion of two more tropic hormones, the gonadotropin's follicle-stimulating hormone, or FSH, and luteinizing hormone, LH. The gonadotropins then act on the testes, stimulating androgen production, the main androgens being testosterone and dihydrotestosterone, or DHT. Hormonal control of spermatogenesis follows long-loop and short-loop feedback pathways. Androgens always maintain negative feedback on gonadotropin release, meaning that increases in testosterone and DHT suppress FSH and LH secretion. Gonadotropins also function in a short-loop pathway, inhibiting GnRH release from the hypothalamus. Lastly, the testes secrete peptide hormones directly influencing pituitary gonadotropin release. Inhibins inhibit FSH secretion, while activins stimulate FS secretion and promote spermatogenesis. The primary role of FSH is to concentrate testosterone in the seminiferous tubules by producing ABP, which, as mentioned previously, prevents testosterone from diffusing out of the tubule. LH seems to be less significant in spermatogenesis, other than its primary role in stimulating testosterone production. Both FSH and LH are believed to facilitate spermatozoa production by preventing apoptosis of developing spermatogonia. So the last step of spermatogenesis involves the differentiation of spermatids into mature sperm cells in a process called spermiogenesis that I will discuss here. This process produces one sperm cell per spermatid, yielding a total of 4 spermatozoa. Spermiogenesis can be divided into 4 stages: the Golgi phase, the cap phase, tail formation, and the maturation stage. In the Golgi phase, the spermatid starts to form a head that will become the acrosome. Spermatid DNA, the chromatin of the nucleus, condenses into a dense structure, and energy-producing mitochondria concentrate in the midpiece of the spermatid body, as you can see in this sperm cell below. The cap phase then occurs as the acrosome flattens out to form a cap over the condensed nucleus. The acrosomal cap contains enzymes essential for fertilization of the egg. Spermatozoa start to develop a flagellated tail, as you can also see here, which helps propel the sperm through the female reproductive tract. It is important to note, however, that sperm do not swim out of the male reproductive tract. They are pushed through muscular contractions. During maturation, spermatozoa lose most of their cytoplasm and unnecessary organelles, known as residual bodies, which are phagocytosed by Sertoli cells. Sperm cells released into the seminiferous tubule lumen are not yet motile and thus not viable. They are transported to the epididymis, where they complete maturation, and they are stored until they are ready to go fertilize an egg. And here are the sources that I used to research the topics that I discussed.